All right, so start off here, guys. Just a quick review of the big three, okay, cellular transport processes, okay, because we talked about those last week. Okay, osmosis, okay, is one of the big three. But remember, osmosis, while passive and requiring no energy, only transports what material? Water. Okay, really the key thing to remember there. That's kind of the dead giveaway on osmosis. Okay, whereas diffusion can transport what? Anything, yeah. Okay, the limitations for diffusion is that it's not going to do, this isn't going to work across a membrane. Osmosis can, active transport can, diffusion will not. Okay, diffusion will work basically within the cell. Everything is transported within the cell through diffusion. All right, and like we said, it is passive. It's moving from high to low concentration, so an area where it's maybe being produced or where it's just present in a large amount, okay, to an area where it's present in lower amounts, basically where it's needed, okay? And remember that it's slow and it's not targeted, okay? We can't say to diffusion, that stuff needs to go over there, right? It's simply not going to do it. It's going to spread it everywhere, right? It's kind of the shotgun approach to cellular transport. Okay, and then with active transport, okay, remember that it's active, it uses energy, okay, it moves against the gradient, so it's going to move stuff from low to high concentrations, it's going to pile stuff up, okay, and it happens also across the membrane. All right, so osmosis and active transport are both across the membrane, whereas diffusion, the one we're going to be most concerned with today, okay, happens within the cell. All right, now we talked a little bit on Friday as well about the effect of a cell getting big on its ability to transport material. All right, and we said that the problem is that everything's transported by diffusion, and the bigger a cell gets, the farther away places within the cell are. Everyone with me there? All right, so what we want to look at here basically is kind of that effect, and I think I drew something like this, okay, on Friday with the two different size cells and showing you kind of how close together everything was. Okay, obviously it's going to take longer for materials to move from one place to another within this big cell if there's only one way to move them. All right, want you guys to quickly. Do you guys have that in your notes package? Just double check. I can't remember if I put that in there or not. If you don't, you'll need to write it down. Okay, so the big thing we got to realize here, guys, is that there's two parts essentially to a cell. It's surface area, and that's essentially where everything comes in and all the wastes go out, okay, and the volume, okay, the internal part of it that has all the organelles and where all the processes go on that needs to be supplied with things and that needs to have its wastes exported or excreted or whatever. Okay, now the problem with this is that the cell membrane is probably the more important part of that because it's the thing that gets everything and gets rid of everything. The problem is that as a cell gets bigger, the surface area increases, but not as quickly as the volume inside. All right, the volume inside increases faster than the surface area because it's three-dimensional, whereas the surface area is two-dimensional. All right, when you calculate surface area, how do you do that? If you're calculating the surface area of a three-dimensional object, you go length times width times, nope, that'll get you volume, times the number of sides. Right? I mean, if it's a cube, it'll be times the number of sides. If it's an irregular three-dimensional object, you'd have to calculate the surface area of each side and add them together. Agreed? All right. Now, when you're calculating volume, you've got length times width times height. All right? This number is always going to get bigger faster than this number because the number of sides won't change. Okay, as a cell gets bigger, its number of sides doesn't increase. Okay, these two increase, but this number remains the same. Okay, with volume, this number gets bigger, and so does that number. So we're always getting a bigger number for volume. You got to remember, a cell is, even though very small, still three-dimensional. Okay, and volume increases very, very quickly. For an organism like ourselves, it's very, very large. Okay, we've overcome that problem by having a system within our bodies that's dedicated entirely to transporting materials to all of our cells. And that way our cells can remain very, very small. Okay, now, does your blood go absolutely to every single cell in your body? No. Okay, but your cells are separated from each other by what's called interstitial fluid. All right, basically it's just bodily fluids that that separate your cells. Now, it's really, really small separation. In fact, lots of times the cells are kind of touching each other, but there's this fluid in between. Material diffuses out of the bloodstream and into this interstitial fluid, and that's where the cells get their stuff from. All right, so when you look at the picture you're going to get on your lab that looks like this, okay, these three boxes are representing a normal-sized cell. So micro is a normal-sized cell. Macro is an extraordinarily large cell. Okay, and multimicro is a multicellular organism consisting of normal-sized cells. All right. Now, people often look at this and say, well, Mr. Coder, that big cell and all of those little cells have the same problem. What if you're one of the little cells that's in the middle of this three-dimensional object? Okay? You don't have access to anything because you're not on the outside. Well, yes, you do. Remember, those cells are not cemented together. Okay? There's not a barrier from the outside to the inside. There's that interstitial fluid that allows materials to get in to the middle. All right? Is everyone with me there? Okay, so the problem we're investigating with this is, how does the surface area relate to volume in cells of different sizes? Does dividing a large cell into smaller cells affect the surface area to volume ratio? So this is the main thing that we want to look at. Basically, what's the effect of a large cell size on the cell's ability to transport things? Okay, so our hypothesis, we got to generate a hypothesis about the relationship between surface area and volume in cells and how that can affect the functioning of the cell. All right, so I would say probably the if part should talk about that. Okay, they should talk about how getting big affects transport. Okay, the and part is that we're going to compare imaginary cells, okay, of different sizes and organizations. And then the then part should probably tell me, what are you going to see in terms of surface area and volume for each of these three things? Okay, and specifically, what we're looking at here is a surface area to volume ratio. All right, that's kind of the way we can make it quantitative, is to look at the ratio. Which one of those numbers do you want to be bigger? Surface area or volume? No. Which one's getting all the materials and getting rid of all the waste? Surface area. And the smaller you are, the bigger your surface area is compared to your volume. All right, as soon as it flip-flops, you're in trouble. Okay, as soon as volume gets to be way bigger than surface area, that cell is in trouble. All right, because now it's got this small amount of ability to acquire things and this huge amount of volume, okay, to take care of it. Okay, the example that we used the other day was, okay, if we had, like, before they built Stony Trail, getting through Calgary was an absolute nightmare. Not that it's, I mean, it's a little better now, but if you stay on Deerfoot, it's still kind of bad. Okay, but when the only way to get from the north end of Calgary to the south end of Calgary was Deerfoot Trail, it was a nightmare. Okay, and as Calgary got bigger, it only got worse and worse and worse. Well, if you're really, really big and you only got one way to get stuff in, you're in trouble. Okay? What? Whereas if you're really, really small, I mean, imagine if we had Deerfoot Trail running through the middle of Okotoks with no traffic lights. Could you get through Okotoks real fast? Yeah. Okay? At 110 kilometers an hour, blink and you'll miss us. Okay? Like, it would be really, really quick. All right? That's what we're looking at here is, as we get bigger and we only have one way to move things or one way to acquire things, okay, we need to make sure that one way to acquire things is significantly large, okay, compared to what we need to supply. All right, so that's the big thing we're looking at, and we should probably mention that in the then part, okay, of our hypothesis. All right, so we're going to have a three-column table, and you can see on the template that I've given, well, actually, you can't see it yet, okay, but on the template I've given you, that's all set up already, okay, so the problem's already in there for you. I did that for you this time, okay. There's no variables to identify this time because we're not actually doing a lab, it's just kind of an activity, okay. You'll have your if, and then hypothesis, okay, procedures here, and you're going to fill in this chart, okay? The cube names, I don't know why I didn't put them in, are micro, macro, and multi-micro, okay? You'll calculate their surface areas, their volumes, and their surface area to volume ratios, okay? And then you'll have the analysis questions to answer, and you're going to need to show your calculations, all right? Now, if you do those calculations on paper, just take a picture of them with your phone and insert that picture, or if you're good with the like equation editor or whatever, okay, you can do that as well, okay? Everybody with me on that one? All right, so now just to give you an idea here, the dimensions of these cubes, because for some reason I thought I had these erased, but when I scanned this picture, they showed up again. These are not the correct numbers. Okay, micro is 1 micrometer by 1 micrometer by 1 micrometer. Okay, its length is 1 because it's a cube. Okay, its height is 1, its depth is 1. Okay, macro is 10 by 10 by 10. And so is multi-micro. Okay, multi-micro is also 10 by 10 by 10. So it's the same size as macro. But it's made up of what? It's made up of many. How many little micros? 1,000. Okay, how do we know that? Because there's 10 micros on a side. Agreed? So the whole thing is made up of 1,000 little micros. And how many sides does each little micro have? 6. Okay, are you seeing the advantage here? All right, this can only absorb material from this face, this face, this face, the one in the back we can't see, the one over on this side we can't see, and the one on the bottom. All right, this one can absorb material through the 6 faces of 1,000 little tiny cells. Is that a bigger surface area? Way bigger. Okay, that's why you have so many little cells in your body. Okay, specifically for that reason. So when we're calculating surface area and volume for these two shapes, it's easy. It's length times width times height. Okay, for the volume, length times width times the number of sides for the surface area. But for this one, if we already know the surface area of micro, the surface area of this will be how many times that number? 1,000. Okay, you guys following me there? So when we're filling that in, that's the math you're going to have to do. It's not terribly difficult math. It's like, what? When did you do that kind of geometry? Grade 7? Something like that, right? Yeah. Okay. All right, so the math's not difficult. Remember that there's 1,000 little micros, okay, in this box here, okay, and their dimensions are 1 by 1 by 1 micrometers. Okay, and obviously a cube has 6 faces. So when you're calculating the surface area, it's, you know, length times width times 6. Okay. All right. Questions on that? Okay. When you get to the analysis, right? The analysis questions are set up like this here, right? For number 1, the wording is sometimes confusing for people. It asks when the cube dimensions increase from 1 micrometer to 10 micrometers. Okay, the increase is a factor of 10, so the sides get 10 times bigger. Okay. But how many times bigger did the surface area get? That's what this question is asking. And how many times bigger did the volume get? So they're asking you here, compare the surface area of macro to the surface area of micro. How many times bigger? How many times bigger is the volume of macro compared to micro? Right? That's what those questions are asking in case that's confusing for you. Okay, for number 2, for the 2 cubes that are 10 by 10 by 10, so the 2 big ones, okay, the size and volume are the same. That's what they're saying here. The size and volume of a factor of 1. They're the same. Okay, calculate the factor for the difference in surface area between the 2 cubes. What that's asking, how many times bigger is the surface area of micro than, or sorry, of multi-micro than macro? All right, so how many times bigger is the surface area of multi-micro than macro? Okay, and for number 3, this is probably one of the more important questions on here. If you make micro bigger, okay, does the surface area to volume ratio change? Well, you can look at that by looking at macro. Okay, if you make macro bigger, does the surface area to volume ratio change? You can do that by trying a sample calculation by making it 100 by 100 by 100. Okay, but for multi-micro, if I make multi-micro bigger, what I have to remember here is that I won't make the cells bigger, I'll make the organism bigger and fill up the volume with more cells. Okay, keep that in mind when you're answering number 3. Okay, and then for the diagram at right, okay, it's asking actually about this one. I think on yours, on the version I gave you, I actually fixed that so that it is actually right beside the question. Okay, so what you'll have to do with these is use the sheet I just gave you and actually measure the boxes. Okay, you'll have to measure those boxes and get the size of their sides, get their surface areas and volumes. There's a little bit of calculating to do for number 4, but it's not difficult. Okay, so if we're looking at box A, okay, we'd measure this side and calculate the surface area of this face, and the back face is the same as the front face. Okay, so you'd multiply that by 2, Find the surface area of this face, multiply it by 4, right? Because all 4 of the other sides are the same. Okay. For this one here, for B, same thing. Find the surface area of the front face and multiply it by 2. Top face, multiply it by 2. Side face, multiply it by 2, and add them all up. Same thing for C. All right. Everybody follow me on that one? Okay. Then you're going to see if cell size, or sorry, cell shape has any effect on the surface area to volume ratio. Because when you do the calculations, you'll find the volumes of these 3 cells are very, very close. Okay? They have almost exactly the same volume. But you're going to find their surface areas are considerably different. All right? And some of them will have better surface area to volume ratios than the others. And remember that that's our quantitative comparison of how well a cell works. If the surface area is big and the volume is comparatively small, that cell's working out very well. Okay? Everyone follow me there? So we're looking at, does the shape of a cell affect its surface area to volume ratio in question 4. Then in question 5, we're looking at real cells like these fake cells. All right? We've got liver cells, skin cells, and nerve cells. Okay? Liver cells are fairly uniform in their shape. All right? They're kind of like cell A. The skin cells are flat, okay, kind of like cell B, but not very long. And then nerve cells, you have nerve cells in your spinal cord that are 1 meter long. One cell. It's 1 meter long. Okay, in fact, you have lots of them. But they're really, really skinny. Right? They're kind of like box C. Right? So we're looking at, does surface area and volume get affected by cell shape, and how have some cells in the body adapted their shape to do their job? Right? Cells in the liver are supplied very well with blood. If there's one organ in your body other than the heart that gets lots and lots of blood, it's the liver. Okay? Skin cells, well, they're only supplied with nutrients on one side because obviously the other side is exposed to the air. Okay? Nerve cells, well, they got to transmit nerve impulses. Probably a good idea to make the cables kind of long for that job. Okay, and then the last two, number 6 is about red blood cells. Okay, and red blood cells have what we call a biconcave shape. So they're shaped a bit like an hourglass. Okay, so they're shaped a bit like an hourglass. So they look like this from the side. They kind of look like a bone. Okay, looked on from the side. All right. Looked on from straight on, they look kind of like a donut because this area here is squished in and the rest of the cell kind of bulges out around it. Now, what that does is, when you have that indentation, which do you suppose has more surface area? A cell shaped like this or a cell shaped like that? Okay, that's what question number 6 is asking. Okay, again, is that shape adapted to do anything? All right, now, there is a sacrifice to this. The red blood cell has no nucleus. Okay, red blood cells do not divide. Okay, cells in your bone marrow and in your liver divide to produce them, but blood cells, once they're shot, die. Okay, they don't reproduce. Okay, so your body has to constantly have cells that are dividing in order to produce them, okay, but they don't reproduce themselves because they don't have a nucleus. All right. Okay, and then the last question there, okay, is about diffusion. That's probably the most important question in the analysis on the whole lab. Okay, so make sure you do a good job of that one. Explain it really, really well. Then your conclusion, and that's it. There's no evaluation here because you didn't actually do any experimenting, so there aren't any sources of error. Okay, questions there? Everyone's good with that? All right. You got all class today. You got, what, another like 50 minutes to work on this. Okay, so you should get a pretty good start, if not done. Okay, computers are right here. It's going to be due on Friday. So whatever you don't get done in class today, you got to have done for Friday.